So, next speaker is Rodrigo Guidoca.
Okay. Can you hear me? Is this working? Okay. So first, thanks a lot to the organizers for inviting me to talk here. And let me start by saying that I think we may all agree that one of the most fascinating things that the brain does is to create meaning from sensory information, from very noisy, ambiguous, and very little sensory information. So we create a meaning where consciousness or some attributes that we extract from what we see, what we hear, what we touch, and so on. And we don't see pixels. We don't see like basic level features. We see people, we see objects, we see a glass, a computer. I see Jorge, Paul, Maria, and so on. So basically, our mind works with concepts. I mean, these concepts is somehow the meaning that we extract from whatever enters our eyes or our ears and so on. So in these 45 minutes, I will make three claims. First, I mean, the main question is, how does the brain encode this meaning? How does the brain encode these concepts? The first claim I will make is that there are neurons in very high-level areas in the human brain that they do encode concepts. This is what we call concept cells. I mean, and other people call it Jennifer Aniston neurons and so on. And I will show you a lot of data to show you that this is the case. I mean, to try to prove you that this is the case. These neurons are really encoding concepts. The second claim will be to say that these neurons encode consciously perceived concepts. So basically, these neurons are responding to what the subject believes he's seeing or hearing or whatever, not what is actually out there. So we did some tricks, some manipulations to show you that the neurons actually do follow the conscious perception by the subjects, irrespective of the stimulus that is out there, that is entering the eye. So that will be the second claim, and I will show you a couple of experiments showing that. And the third claim will be more like an hypothesis, and hopefully I will finish there. I mean, in time, and we can have an open discussion about it. I will argue that these neurons have a lot to do with forming and recalling memories. I will argue that this is the main function of these neurons I will describe, and I will show you a very, very simple model of how I believe this is done, and then we can discuss it, I mean, as long as you want. Okay, so let me start with the data. I mean, let me start by showing you that there are neurons in the brain that encode concepts. So first of all, I will talk about humans, and I will talk about single cells. So the first thing I have to clarify is, well, how can we do that? Because you know that to record single cells, you have to insert electrodes. This is typically done in animals. But there are very specific cases where such recordings are done in humans. So this morning we heard about one of these cases where, I mean, you have intracranial recordings. Another typical case where intracranial recordings that are in humans are in patients suffering from epilepsy, that they are subject to epilepsy surgery. Basically, they try to extract the area that starts the epileptic seizures to cure the patient. And these patients are typically, or in many cases, these patients are implanted with intracranial electrodes, and this is not working. Well, they're implanted with intracranial electrodes, and you see there the surgeon implanting the electrodes, and this is done since decades. It's quite a standard procedure, and the idea of these electrodes is to try to localize where the seizures come from, where the epileptic concept is, so because this will be the area that the surgeon may potentially target. Now, these are the electrodes that are typically implanted, and I don't know, someone knows how to. Doesn't work. Well, basically, these are the electrodes that are implanted. This is quite typical. These are what is called the depth electrodes. They're about 1 millimeter thick, and they have several contacts. This one's here. And from these contacts, you can record intracranial EEG. So you can record EEG very local to the tissue surrounding the electrode. Now, something that was developed at UCLA, particularly by Itzhak Fried in the 90s, was to put these microwires coming through the electrodes and popping out at the end of the electrode. So basically, thank you. Thanks. So basically, this is the clinical. So these are the clinical electrodes, and these here are the microwires. So this is quite standard. This is done in many, many centers, but what was quite unique at UCLA is that Itzhak developed a way to put microwires going through and popping out of the tip of the electrode. And these microwires are kind of similar to the things that people use to do animal recordings. They are very, very thin. I mean, they are 40 microns thick, and there are 8 microwires coming out of here, and then there is one reference. So these microwires allow us to record single cell activity, many, many neurons, and we can individualize. I mean, we can tell which neuron is which. All the data I will show comes from these microwires. I will not be talking about clinical contacts. Now, where are these electrodes implanted? Well, it depends on the patient, depends on the clinical case. But it happens very often that these electrodes are implanted in the medial temporal lobe, which is the hippocampus and the surrounding cortex, entorhinal cortex, parahippocampal cortex, amygdala. So we have recordings there of single neurons. And before I show you any data, I want to refresh you this slide, which I guess all of you have seen many, many times. And 99.9% of the times that you saw that is just basically to say how complex the visual system is. The 0.1% will be just to say we're recording here, right? So this is just to make it clear that there's nothing magical with these. I mean, there's nothing special about these neurons. They just happen to be on top of a very, very heavy processing done in earlier areas. I mean, it's not that something come out here out of the blue. Well, there's a lot of processing going on here. And you know that in previous areas, in inferotemporal cortex and so on, you have very, very high-level representations like faces, body parts, and so on. So you can expect that here, hippocampus and entorhinal cortex, you also have a very, very high-level representation of, in our case, visual stimuli. Now, data. That's the first recording. It's a very nice, clean recording because it's also responding to somebody you may know, Maradona. So, and this is basically just the recording coming out of one microwire high-pass filter. The only thing I did is high-pass filter the data. This is what you see. So you have noise. So, and the experiments we do is basically showing pictures in the laptop. I will go with my laptop, put it in front of the patient, and I will just show pictures. And I will show them for 1 second, the next picture, and so on. And the pictures, I mean, I show many times the different pictures, and are all randomized. I mean, they're all randomly intermixed. So basically, this neuron didn't fire to a picture of Brad Pitt, but fired very strongly to a picture of Maradona, the football player. Didn't fire to another sportman from Argentina, a basketball player, Manu Ginobili. Didn't fire to Robert Plant, Led Zeppelin, and it fired again to the picture of Maradona. So there are very strong responses in the medial temporal lobe. This neuron was actually in the amygdala, and to visual presentations, to visual pictures. I'm still not saying what the neuron is firing to. This is what we are going to explore now in the following slides. But there are very, very strong visual responses. Now, you never get something like this. This is a very, very unique case. Typically, what you get is something like that. Because you don't record from one single neuron when you have a microwire. If you do animal experiments, you may, because you are moving the electrode until you get very close to the neuron. If you have chronic recordings, well, close to the tip of the electrode, there will be many neurons. So and this is what the data will typically look like. So, but you can do some mathematical trick called spike sorting, and you can separate the firing of different neurons based on their shapes. So you know that different spike shapes correspond to the firing of different neurons. So these are all the spikes you detect, and if you do clever signal processing called spike sorting, you can tell, well, you have one here, one here, one here, one here, one here. Now, if you see the responses of these spikes, of this recording, to different pictures, just the detected spikes, this is what you see. This is time 0 here. We have 1 second baseline and 2 seconds response. There's really nothing. You don't see anything. But now if you see the responses neuron by neuron, not just the detected spikes, but you separate neuron by neuron, look what happened. This is this neuron here. This neuron fired very, very strongly to Vladimir Putin. And I will show you now this neuron here. This neuron fired very strongly here to the Taj Mahal, to the picture of the Taj Mahal. Now, this makes things quite challenging because if you don't do a good job in doing this, you don't see the neuron. I mean, if you see them all together, if you're not able to separate these neurons, if your spike sorter is not good enough, you don't see them. That's why it's hard to see these neurons. But we have a good spike sorting, that works. That's fine. But this is the second more difficult point. The second more challenging point is that if I wouldn't have shown the picture of the Taj Mahal, I wouldn't have seen this neuron. Because the neuron was nearly silent. In baseline, it was nearly silent. It didn't fire to all the other pictures. If I wouldn't have shown the picture of Vladimir Putin, maybe I wouldn't have seen this neuron either. This neuron either. Because it's firing mainly in response to Vladimir Putin. If I wouldn't have shown the picture of Maradona, I wouldn't have seen the neuron from the previous slide. And I'll show you we're in Italy, so I'll show you an example. Oh, you cannot see it. This is a map of Italy. So this is a picture of a map of Italy. Whenever I show the picture of the map of Italy, the neuron fire, this is baseline, no? From 0 went up to 40, 50 hertz. But look at the responses to other pictures. I show about 100 pictures. It didn't fire to any of them. So if I wouldn't have shown the picture of the map of Italy, I wouldn't have seen the neuron. The neuron would have been there, but I wouldn't have make it fire, I would have missed it. So the question is, how do we know which pictures we should use to make these neurons talk, no? to get them to fire and to see them? Well, we don't know. But what we do is we split the experiments, and we first start with what we call a screening session. And the idea of the screening session is, well, let's try to show as many pictures as we can, I mean, with time constraint, because we don't want to bother the patient too much, about half an hour. And let's see if any of the neurons fire to any of the pictures we are showing. So this is what we call a screening sessions, and I'll show you one example. We call them also AM sessions, because we tend to do these sessions early in the morning, see what we have, and then follow up with other experiments. So this is one neuron in the hippocampus of one patient, more than 10 years ago now. It didn't fire to any of these pictures here, didn't fire to any of these faces, but it fired very strongly to Jennifer Aniston. Right? That was the first neuron I got doing this. I will show you in a second what this neuron did. That's why people started the Jennifer Aniston neuron. So it fired very, very strongly to Jennifer Aniston. Now, you may wonder, well, wait a second. How do you know it's firing to Jennifer Aniston? Maybe it's firing to something specific about this picture that is not in the others. Well, I didn't show just 15 pictures. I show about 117 pictures in this session. So I show many, many more, right? But you can still argue, well, maybe there's something of this picture that is not in the others. Maybe the blue background, maybe the contrast of her hair color with the blue background, or something like that. Now, the trick is that we can analyze the data very quickly. And within an hour or so, we can see this response. Or maybe around an hour after doing the experiment, we can see this response. So then what we did was very simple. Once we saw this neuron firing to Jennifer Aniston, we went to Google Images, get many pictures of Jennifer Aniston, and repeat the experiment and see what the neuron did. So, and we show, of course, many pictures of other people as well. No, we want to have controls. So the hypothesis was, or the question was, are these cells firing to some visual features, like the blue background, or they're really firing to her, to Jennifer Aniston? So let's see what happened. We repeated the experiment shortly after, and this is what the neuron did, the same neuron in the hippocampus. It didn't fire to, actually, the Eiffel Tower with a blue background. It didn't fire to other people. Another blonde actress, Pamela Anderson, if you want to know. I mean, another guy, guys, different guys. And these are 3 different pictures of Jennifer Aniston, and the neuron fired very strongly. I showed you more responses of the same neuron. Another picture of Jennifer Aniston, the neuron fire. Another picture of Jennifer Aniston, the neuron fire. Different pictures of Julia Roberts. The neuron didn't like it. I mean, another famous actress. The neuron didn't fire to Julia Roberts. Didn't fire to Kobe Bryant. Didn't fire to many other people. It was basically firing to Jennifer Aniston. Again, another picture of Jennifer Aniston, the neuron fire. Another picture of Jennifer Aniston, the neuron fire. This I named it in passing. If you want, we can discuss it later. It was quite funny. Then it became quite intriguing. I think I have an explanation for it. You put Jennifer Aniston with Brad Pitt. At the time they were married, the neuron didn't like it. The neuron didn't fire. We can discuss it later if you like. So basically, this is a summary of the whole session. I show 87 pictures, and the responses in red, these are the mean responses, are to the pictures of Jennifer Aniston, which are the ones that I show here. So basically, the neuron prefers the picture of Jennifer Aniston, very different from each other, right? As different as I could get them in the little time I have to do a Google search in Google Images. Okay, let me skip that. So another example from another patient. We have tons of these examples. I just show you a couple. This neuron fired to Halle Berry, another famous actress, right? Same trick. We say, well, is it really firing to Halle Berry? Let's go to Google Images, get many pictures of Halle Berry and pictures of other people, and repeat the experiment. So this is what we got. So no response to Paul McCartney, George Bush. I don't know who they are. This is Halle Berry. That's a good response again. And another Halle Berry, another Halle Berry, another Halle Berry. So 4 pictures of Halle Berry. So you get 4 good responses. Again, you show Julia Roberts, Kobe Bryant, other people, the neuron didn't fire. It fired just to Halle Berry. I'll show you more pictures of the same neuron. Actually, that was very interesting. There was something very interesting happening at the time we did this recording. Halle Berry was releasing a film called Catwoman, which sounds like stupid to say, but it was fun because we could show the picture of Halle Berry dressed as Catwoman, and the neuron also fired. The patient actually knew that that was Halle Berry, right? And the neuron fired as good as if you show the picture of Halle Berry. Again, not firing to any of these people and so on. We try cartoons, caricatures, because the idea is to distort the features of the face and see if the neuron will still fire. And we try different cartoons. Here is Eddie Murphy, for example, and here's the one of Halle Berry, the neuron fire again. So basically, I have many of these examples, and the point I'm trying to make is what I told you at the very beginning. I want to show you that the neuron fired to the concept and not to any specific features of some particular pictures, right? Now, these experiments are done in humans, so there's a very easy and very conclusive way of making the point. And the very easy way to show it is actually to write the name of the person and to see what the neuron does. So this is what we did. So basically, in the experiment, we intercalated pictures of different people, and every now and then will appear one name. And I show you all the names together. This is the same neuron. The neuron didn't fire to, I don't know, Kobe Bryant, Julia Roberts, I don't know what it says here, George Bush. No, actually here is Catwoman, George Bush, and this is Halle Berry. So basically it's the response to the letter string Halle Berry in my computer screen. So it's a very strong response, as good as when you show the picture. So it's really firing to the concept. There's no visual features here. It's just a of Halle Berry. Now, what about sound? What will happen if instead of writing the name, I will say the name? Or I will get my computer to say the name? Well, it seems to be the same, no? Because if it's firing to the name, to the written name, it's firing to the concept. Well, but the spoken name is a different sensory modality. So the question was, will these sensory modalities meet at the levels of single cells in the hippocampus? And after this long introduction, you can expect the answer. So this is another neuron in the hippocampus of another patient that fired very strongly to Oprah Winfrey. Look, this is time 0. This is when we show the picture. This is 1 second baseline. There was no spike in no of the trials preceding the picture of Oprah Winfrey. But then you get a huge response, up to 50 hertz, when you show the picture of Oprah. Another picture of Oprah, another response, another picture of Oprah, also a good response. This is the letter string, Oprah Winfrey. You get a good response. And this is just the response to, this is the response to my laptop saying Oprah. Let's see if you...
Oprah.
That was a stimulus. That was it. And the neuron fired very, very strongly. And if the laptop say...
Cameron Diaz.
It's a computer synthesized voice, no? The neuron didn't fire. So it's multimodal. So there's a convergence of different sensory modalities. Now, another very interesting example, very, very interesting. The most interesting of, well, one of the most interesting I will show you, because this is Luke Skywalker from Star Wars. But there's something very interesting happening with this neuron. So that was in the entorhinal cortex of another patient. It fired to the letter string Luke Skywalker. Again, look at the baseline, 0. You show Luke Skywalker, and there's a very strong response, from 0 up to 40, 50 hertz. So it's very, very brutal, the change in firing. This is the letter string Luke Skywalker, very strong response. And this is Luke Skywalker being said by a male and a female voice. And I show you this.
Luke Skywalker.
So that was this sound, this is the response, and now this one.
Luke Skywalker.
Is this response, right? So fire very strongly.
Keanu Reeves.
And when the computer says Keanu Reeves, nothing happened. So do you see something interesting going on there in this neuron? Look at this. You know who this is? Well, you know Star Wars, right? Yoda. This is another character from the same film that, contains Luke Skywalker. So they're both characters from Star Wars, right? The film. So, and this is very, very interesting. And this is the first clue to the interpretation I will give to this data. We have many neurons like this now, and we did experiments to show that this is the case. It happens every now and then, or actually it's not every now and then, it's quite common that neurons, like the ones I'm talking about, may fire to more than one concept. But these concepts tend to be related. It's not just something at random, they are related concepts, like Luke Skywalker and Yoda. Okay, this is another very interesting neuron, very, very interesting. You don't know this guy, or you may know him. So this is Arne Engstrom. And this is a guy that was doing experiments at UCLA. He was actually studying place cells and other stuff. This is interesting because the neuron fired very strongly to 3 pictures of him, to the name written on the screen, and to the name saying in the computer. And the interesting thing is that the patient didn't know Arne a couple of days before I did this experiment. So basically, this shows me that this conceptual representation in the hippocampus and medial temporal lobe can be created relatively quickly. It can be a matter of days or even seconds. It doesn't take months or years, right? Okay, let me skip this. This is basically just to make sure you are not blaming me for saying these are grandmother cells, because they are not. I want to make it very clear that these are not grandmother cells. Now, let's go slowly. Why are not grandmother cells? There are many different ways of you can define a grandmother cell. The most extreme version is to say there's one neuron per concept. So there's one and only one neuron, encoding Jennifer Aniston, at least in this area. This cannot be. It cannot be based to this data, because if I did find a neuron firing to Jennifer Aniston, there have to be more. I have to be extremely lucky to find the one and only among billions of neurons in this area, right? So I just found one of many encoding for Jennifer Aniston. You can still say, well, maybe you have a few thousand neurons, or maybe 1 million neurons, encoding to one and only one concept, right? This is in principle possible. It is very hard to prove, because I cannot show every possible concept around. But I do have many cases. I show you one, and I have many more, where this neuron fire to more than one concept. So this is actually not the case. It can happen in some cases that I find one neuron firing to one thing, like Halle Berry, the Halle Berry neuron. But maybe if I would have shown more pictures, I would have found that this also fires to something associated. So I don't believe these are grandmother cells. But they are extremely selective. So they don't fire just to half of the pictures or so. They fire to very, very few of the things I'm showing. So it's a very sparse code. They fire to concepts and not to details. They fire to the name of the person also. And if they fire to more than one concept, these concepts tend to be related. And that's, we did some experiments where showing that this is also very, very clearly the case. Now, do these neurons follow the second point now? I hope I convinced you that neuron fires to concepts. Second point, do they follow conscious perception? So we did a very simple trick. We flash the image, and then we see what happens if in some cases the patient will see the picture, and in some cases will not see the picture. So imagine I have the neuron firing to Halle Berry. What will the neuron do if I flash the image and the subject sees the picture? And what will it do when I flash exactly the same image, exactly the same time, and the subject doesn't see the picture? Will it fire the same way because it was exactly the same stimulus? Or will it fire in a different way because it was a different perception? In one case he saw it, in one case he didn't. So we show, to try to get this threshold of perception, we show the pictures with at different times, with different durations. So going from 33 milliseconds up to a quarter of a second. This is very easy to see. This is very hard to see. But in this case, the subject saw the picture in all the trials. And the response to, in this case, the neuron firing to Whoopi Goldberg was the same for all durations, was very, very similar. So duration of the picture didn't matter, really. As long as the subject recognized it, the neuron fired. Let me show you another example, another neuron. This one fired to the brother of the patient, a picture of the brother of the patient. Now, look how interesting. If I show the picture for a quarter of a second, the neuron fired in all the trials. The subject saw the picture in all the trials. All these blue marks mean that the subject saw the picture. The subject said, I recognize the picture. So he pressed the Y key. All these trials, the subject saw the picture, except in this one. In this case, he said, I didn't recognize the picture. He was looking at the screen, no? But he didn't recognize the picture. The neuron didn't fire. And this is the interesting one. In these first 5 or 6 trials, the subject said, I didn't recognize the picture, although he was looking. The neuron was completely silent, as if the picture was not there. In these other trials, it was exactly the same stimulus, exactly the same duration. The subject recognized the picture, the neuron fired nearly as good as when you show it for 1 second. I have plenty more of these examples. This is another one. This is a neuron that fired to the former World Trade Center. So 33 milliseconds, the subject didn't see the picture in any of the trials. The neuron is completely silent. Not even one spike to not even one of the trials. Nothing, 0. You show the picture for a quarter of a second, there's a very good response, and look here. You show it for 66 milliseconds. These first 4 trials, he didn't see it, complete silence. Then he saw it, the neuron fires to top. And then the last one, he didn't see it, the neuron silent again. So there are many examples of neurons that they have this kind of like all or none responses going hand by hand with perception, what the subject sees. Okay, I show you one more experiment. This is the newest of all. I think this is the last piece of data I will show you, and then I go to the third claim about these neurons doing memory. So what's a similar idea, no? We want to see if, how tightly the neuron follows the visual input against the subjective perception. So we try to manipulate that. One way of manipulating this is by morphing. So for example, here we have Angelina Jolie, and here we have Halle Berry, and with a different degree of morphism, and then we come up with an ambiguous picture that sometimes you may see as Angelina Jolie, sometimes you may see as Halle Berry. So the trick is, for example, if we do have a Halle Berry neuron, what will the response be to this ambiguous picture? Will it be the same when the subject say was Angelina Jolie to when the subject said it was Halle Berry? The picture was exactly the same, right? And we used psychophysics tricks, we used phase adaptation to try to bias the perception a little bit. And let me show you a couple of examples. That was done by Sasha Kraskov, the experiment. So that's the most horrible morphing you will see ever. So that's a morphing between, well, I had a very good response to Whoopi Goldberg. Who can you morph Whoopi Goldberg with? Well, Bob Marley, of course. So the neuron fire very strongly to Whoopi Goldberg, and this is the morphing between Whoopi Goldberg and Bob Marley. So it's quite different to Angelina Jolie and Halle Berry, I would say. So basically, the neuron fired to Whoopi Goldberg, but not to Bob Marley. And look at this. This is the response to this hybrid picture, but separated according to the decision. This is when the subject said is Bob Marley. There's no response. And this is when the subject said is Whoopi Goldberg. Look, it's a very strong response. It's exactly the same picture, right? Now, there's something more interesting here, or one more interesting piece of information. Every now and then, instead of showing the morphing, we show the full picture, the original picture, like this one here. And this is what I call 100% picture presentation. Now, when we show the original picture, in this case, when I show the picture of Whoopi Goldberg, the response was the same as when I show the morph picture, as long as the subject said it's Whoopi Goldberg. Quite different than showing this picture and the subject saying it's Bob Marley. So it doesn't matter what you show to the subject. It matters what the subject believes he's seeing or believes what is out there. So we have tons of these examples. I mean, it's a morphing between Seinfeld and Orlando Bloom. And again, this is the ambiguous picture when the subject said it's Seinfeld. The neuron did fire to Seinfeld, but not to Orlando Bloom. The neuron fired more than when the subject said it's Orlando Bloom. So let me show you the population average. So let's stay with this one. I mean, this is more or less giving the same information, but let's stay with this one. So all the pictures for which the neuron didn't fire originally, I put them together as picture A. All the pictures for which the neuron fired, like Whoopi Goldberg, I put it as picture B. So this will be Bob Marley. This will be the average of Bob Marley or Orlando. The different neurons didn't fire, population average. And this will be the average of the pictures for which the different neurons fire. So we normalize the population average, get the response strength. And you see, this is the response to the morph picture, where the subject said it is the picture that the neuron liked, and when the subject says the picture the neuron didn't like. So that would be, say, Whoopi Goldberg or Seinfeld. That would be Orlando Bloom or Bob Marley. So there's a huge difference. And this is exactly the same picture. Now, the response to the morph picture when the subject said is Whoopi Goldberg was the same as if you show Whoopi Goldberg. Similar strength. And the response the other way, when the subject says the non-responsive, was also statistically the same as when you show the full picture of the non-responsive one. So the neurons do follow what the subjects believe they're seeing. Okay, I finished by proposing a function of these neurons and giving you a very, very simple cartoon of how I believe this function is accomplished. So, and to discuss the function of these neurons, I have to start by talking about this guy. I cannot ignore the evidence, huge evidence, about the function of the hippocampus and the middle temporal lobe given by HM and many other experiments that follow in the finding with HM, right? So, basically, we know, I guess you all know who HM was. He had a surgery that removed both hippocampi, and then he became amnesic, right? So he developed anterior grid amnesia. He couldn't form new memories. But the perception of HM, visual perception, was okay. HM could still recognize people he knew from before the surgery. He could recognize objects and so on. So basically, if HM didn't have hippocampus and middle temporal lobe, and he could still recognize things, well, the neurons I'm reporting here, they're not involved in perception, because we know that you can take them all out and you can still see, you can still recognize things. So they're not involved in perception. They're actually not the final storage of memory, because HM could still remember things from the past. HM knew who his mother was and who people he knew from before the surgery, right? He knew who the people from before the surgery. So it's not the final storage of memory, but the hippocampus is crucial to form new memories. If you don't have the hippocampus, you can't form memories anymore. And this is what we know from this evidence and many other cases and animal studies and so on. So the proposal I have, and I didn't show you data for that, I will leave it as an hypothesis, and if you want we can discuss it in the discussion, is that these neurons are involved in forming memories. And the way I like to call them is the building blocks of memory. So basically, you have a semantic representation of different concepts, right? You have Halle Berry, you have Jennifer Aniston, you have, I don't know, Luke Skywalker, and so on. So these neurons are there. They are representing different concepts. And the skeleton of memories, declarative memories, and more specifically episodic memories, is making associations between concepts, right? I met Jorge, I don't know, we went to the Lago Garda. Then I'm making an association. My concept of Lago Garda, my concept of Jorge, I make an association between these two concepts, and this is the skeleton, at least, of a new episodic memory. That's how I store it. So my claim is that these memories are stored, are there to create associations. So these conceptual representations are actually the building blocks to be able to create these associations. So what I'm saying is, the first claim is that these neurons, they have an abstract, explicit, and very, very sparse representation of the meaning of the stimulus. Doesn't matter what is out there, what it matters is what the subject believes he's seeing or hearing. And they are the building blocks of memory. And let me tell you how I believe this works. This is the final slide, and if you want, we can discuss from here. So I will use the example of the neuron I showed you before, right? So imagine that you have a network of neurons firing to Luke Skywalker in the hippocampus. These neurons are representing Luke Skywalker, right? And you have another set of neurons encoding Yoda. And imagine, in principle, just imagine, in principle, these concepts are not associated. There are two different concepts. They have two different assemblies representing them. So whenever you show Luke Skywalker, this neuron fires. Whenever you show Yoda, the neuron fires. Now, you create an association. You say, no, these two guys are quite related. You create an association. So what's happened? How can this be encoded by these assemblies? Very easily. Some of the neurons that encoded to Yoda will start firing to Luke Skywalker, and some of the neurons that encoded to Luke Skywalker will start firing to Yoda. So if you have two separate assemblies, the way to create an association is to create an overlap between the assemblies. Some neurons firing to one will start firing to the other. A partial overlap, not a full overlap. If you have a full overlap, then you fuse the two concepts. You cannot distinguish them anymore. As long as the overlap is partial and not that high, you can still have a distinct representation for both things. Now, if you have this, well, then these concepts are now related. The association is encoded here. And the association can be Jorge and the Lago Garda, right? So wherever I see Jorge, I remember being at the Lago Garda, right? So, and that's the basic idea. If I show the picture of Luke Skywalker, if I show you Luke Skywalker, you may think of Yoda. Basically, the neural network encoding for, or the assembly encoding for Luke Skywalker will fire. Some of the neurons will be also firing to Yoda, and then the Yoda network will fire. So that will create a way of encoding, having recalled the flow of consciousness and so on. So and with this, I want to finish. I want to mention, well, these are the guys actually doing a lot of the work, and most of what I show has been done in collaboration with Itzhak and Christoph Koch. Okay, thank you.
Okay, I already have 4 questions. First one over here.
Thank you, it's very nice. I want to ask if you're thinking about all these associations between the concepts that you're presenting. Do you try to follow, let's say in a future patient, or asking them what are the associations that arise from the screening process? So for example, if you see not to start thinking of your ideas or, Googling up some stuff, but asking them what would be the association, like Jennifer Aniston, what are your associations? And then, because it could be that the, friend's apartment, without, a photo of that would, or the music, the clip that comes. So it's a suggestion to try to follow their associations and to see if that could actually substantiate your idea.
So the experiment we did is, let's say that the first step going in the direction that you say, the first thing we did, which is very easy to do, and it's a pity not to do it. And we did it, and it's still unpublished. But it's a very strong result. We asked the patients to rank the associations. How much you relate this picture to the other? How much you relate Luke Skywalker to the other? Luke Skywalker to the Pope? Luke Skywalker to this? And all the pictures with each other, or a selection of pictures with each other. Now, what we found is that whenever we have a neuron firing to more than one picture, for the subject, these pictures were very, very highly related compared to the rest. Now, what you're saying is one step further, which is basically exploring this space of association. This is harder to do because we have a very limited time, and we cannot keep these neurons for long, because sometimes they may have a seizure in between, and the electrons move and we cannot do it. But we are thinking of experiments like this. The third bit of information I can give you is that sometimes we're not just exploring the association space of the experiment because it's very high, but we're trying to create associations, and we're following the neuron. And the way we create association, this is a paper that's under revision, is doing something like this. So for example, if I have a neuron firing to the White House, I do kind of like, I show Matthias, this guy. Matthias showed a selfie, something looking like a selfie of Julia Roberts with the White House. And what he found is that the neuron firing to the White House, then it starts firing to Julia Roberts, the moment the subject remembers the association.
I was especially interested at the fact that the cells fire to the auditory stimuli, not because they're in a different modality, but because they are representations that arise over time. And so we know from psycholinguistic work, for example, that multiple meanings as you utter a word are activated. And I wonder at what point the concept cell becomes activated. So you had Oprah, for example, and when you said, or when your computer said Oprah, the cell fired. What if the computer had gone on to say, not Oprah Winfrey, but Oprah Karamatsa? Does the cell never fire? Does the cell start firing and then get really disappointed and stop firing? Or does the cell say even better?
I do a prediction. Well, I can tell you what we found, and I can do a prediction. I don't know who this second Oprah is. So if you will do the experiment with me, and you will say Oprah, the Oprah Winfrey neuron cell in my head will immediately fire because there's no other Oprah. Now, if I know many other Oprahs, then I will have to wait to know exactly what are you referring to. So what we do find in the responses to auditory stimuli is that the onset, I passed by the slide if you want to see it, but the onset of the response for the pictures and for the text, for the letter string, is nearly the same. It's about 300 milliseconds after stimulus onset. The sound presentations have an onset that is a bit later and more variable. And this is because what you say, and also within a name. If I say, I have a neuron firing say to Arnold Schwarzenegger, until you know who I'm talking about, well, maybe a couple hundred milliseconds happened. And that's why you have a lot of variability in the onset.
But so do you think even if you only know one Oprah, you could still hear the phrase Oprah Karamatsa? What does that cell do at that point? So you decided it was Oprah.
I think the cell will start firing to Oprah Winfrey. If you only know one Oprah, you say Oprah, and the moment you say Oprah, my Oprah Winfrey neuron starts firing, and then you say the surname, and say, oh, God, it's something else, so maybe this will block the first neuron. But I'm speculating, I don't know. By the way, there's something like, I don't know if you're interested in this, but this mechanism can also explain, because what is really, really striking is this RNA neuron. After a couple of, the patient didn't know this guy at all. The face and the name, nothing. And then 1 day later or so, I have a neuron that is firing to the faces of the different pictures of the guy plus the name. So this mechanism of partial overlapping networks can also, explain this very quickly, because maybe the name will be encoded in one assembly, the face will be encoded in another, and when you learn this is the name of this guy, well, you just do this full overlap, and then it becomes the same concept.
Okay, first, I have already a list of 10 hands, I think.
So with these cells that are, you think, building these associations or concepts, you already mentioned that you've started running a study where you're trying to build in this association. But how fast do you think that can actually be built? Because, there has to actually be the forming of physical connections for these to change. Yet you say you're seeing this on the order of seconds, which is a little fast.
Yeah, on the order of one trial.
On the order of one trial, you're able to see a change in the cell's response.
In the experiment that I show you, the moment the subject remembers, so the trial the subject remembers this association, and could be the first time he sees it, the neuron will start firing to the associated concept. Not all of them, but a reasonable proportion in one trial. Which is key, because if you don't have this, I cannot be talking about episodic memory, because episodic memory is a one trial thing. You are not having the experience 10 million times to create an encoding of this. It's one time.
How is that? How do you think that's being instantiated, though? Because that's too fast for new connections to have formed.
Well, the
physical connections could not have changed in that amount of time.
Well, it's synaptic plasticity. The connections are there, but they're not active. So basically in the hippocampus, in CA3, you have random connectivity, but not all of this connectivity is actually functional. So basically you have the connections in there, and what is key in here, I mean, modeling works, especially McLennan and others, have shown that if you have this type of representation, very sparse, then with models you can create very quickly associations. If you have distributed coding, you can't. But with this type of representation, you can.
Okay.
Yeah, I think she was first, yeah.
Hi, hopefully my question makes sense. So I have two kind of questions. My first would be, would you expect like associations like Hitler and mean, like those kinds of associations to also
Hitler
be
and
shared?
what, sorry?
Like associations with traits, like mean or nice or happy or sad, because these are also important associations that are part of our episodic memories.
Yeah.
My second part of my question is, how does this kind of a model differentiate between different kinds of associations? So like parent, child, married, or would that be higher order and somewhere else maybe?
I think all the
or owns or likes or those
yeah.
kinds of things.
I think all the associations you mentioned, they don't happen here, they happen in cortex. So basically what you're doing, you're describing a hierarchical system, a clustering system. Hitler is a very nasty guy, he's very mean and so on. So you are classifying this guy. I don't know, Jennifer Aniston is an actress, is American, is blonde, but all these classifications, they happen in cortex, I believe. Basically, what you want to have in the hippocampus is something that you get rid of all this hierarchical thing, because this makes things harder. I mean, you don't want to be moving hierarchies around. You just want to have the concept there floating and being free to be linked to whatever. And then how you consolidate this in cortex, well, this will take time, but this is consolidation. That's a slower process, but you need a fast process to create a quick association.
Okay, I think Alex.
So it's beautiful data, but I really think that calling these concept cells is really confusing. For the simple reason that the amnesia data, as you pointed out, tells us that all that conceptual work and representation had to go on before information reached the hippocampus.
Yes.
Number one. Number two, what's going on in the hippocampus, I think the sparse encoding idea has a lot of validity and a lot of evidence behind it, but certainly the creation of episodic memories is not limited to pre-existing representation. You can go into a museum and see modern sculpture that you have never seen before. You can remember that. So there couldn't have been pre-existing representation, and I assume that how those episodic memories are being created is very similar to the way you're talking about these cells. So talking about them as conceptual cells usually suggests that it has a direct role in either the creation of conceptual representation at some level, and I don't see any evidence for that. It all happened.
I don't have an evidence of the creation. I mean, I'm limited. I mean, I cannot. Well, we're trying to do experiments for that. But let me answer the 2 points. The first one, I agree completely that there is a conceptual representation in cortex. It's different. I think it's more distributed. It's not that sparse. And maybe you don't have an explicit meaning as you do have here. But there is, of course, a representation of concepts in cortex. I mean, HM knew who his mother was and so on.
Isn't that explicit meaning? Didn't HM have explicit meaning?
Yeah, but it's a network property which is distributed. I mean, explicit meanings at the single cell level. If you get one neuron from IT, a phase cell in IT, you cannot tell who this person is. If you get a single cell from hippocampus, well, you are much better off telling exactly who the person is. In this sense, it's explicit at the single cell level. At the population level, of course, you can tell in both cases. In one case with a distributed coding, and in one case with a sparse coding. So I don't deny that there are concept representation. I'm just saying there is an extra representation of concepts at a higher layer which has the best properties to create associations, right? But I'm not denying the other. Now, in terms of creation, typically you will form memories about things you care about. If you see a person you never met before drinking water, I mean, there's nobody there, no? But if you see a person drinking water that you don't know, you won't remember this because there's no saying. Now, if you see, I don't know, somebody you know very much drinking water, you may create a memory of this, but this person you know, so you may have some representation there. This doesn't deny you can create new concepts like Arne and these things. This guy was unknown to the patient, and suddenly the guy becomes known because he's talking to the patient like, on a daily basis for at least a couple of days, and suddenly Arne becomes relevant, and then you have a network encoding for Arne because he becomes relevant, and then you can link it to other things to create new episodes.
I could argue about that, but I'm not going to take any more time.
We first Mel, then in the back. Did you have a question, Alfonso, as well?
Was it ever the case that the neurons that you were recording from were removed surgically? And if so, did the person lose the concept?
The limitation we have is that we don't interact with the patients before or after the time where they're implanted with the electrodes. So we have,
No, I didn't mean recording. I just meant if you asked them about Jennifer Anderson, would they...
I never saw a patient after the surgery. I never talked to them. I don't think, well, because of ethical reasons and so on, I think in the protocols, we don't bother the patient before or after.
No,
and if we want to do it, we need to change the consents and so on.
Right.
But I have a strong intuition that there's no one Jennifer Aniston neuron, and it's not in one cluster. So this is... So this I can tell you about data.
So these are widely distributed networks in your point,
yes,
from your point of view?
It's sparse, but spatially distributed.
Right, and your evidence for that is?
The evidence for that is that if you do spike sorting, sometimes you get responses. So the Halle Berry neuron had a neuron nearby that I could separate as a spike sorting. I was found to Mother Teresa of Calcutta. No relationship between these two things at all. And this is a very, very common finding. You do spike sorting, you separate neurons that are next to each other, and they're completely unrelated.
Right.
So basically, they're all spread out.
You don't know they're spread out. You just know there are a bunch of different ones in one place. It could be that there's no other Halle Berry neuron miles away, right?
Yeah, they're not.
It could be. So you don't know.
Well, I don't remember seeing. The problem is it is so sparse. The evidence that you're looking for, I think, is for me to tell you, well, I have a neuron in the left hippocampus firing to Jennifer Aniston and another one in the right hippocampus.
Sure.
But the selectivity is so high that the chance of getting two neurons firing to the same.
No, I totally get that you can't find that, but it may indeed be possible that the networks are rather small, not distributed.
Okay, Alfonso, then we have one in the back, and Sharon. And
okay. My question is similar to Alex, but from a slightly different angle. First question is, are these cells responding only to individuals, or do they respond to kinds? So they respond to chairs. And if they only respond to individuals, why? The second part of the question is in terms of your model, where you showed us a possible architecture where you have a number of cells responding to one individual, but then also responding to different individuals. Now, this could either happen by accident, by chance, or it could happen because of intrinsic properties about the individuals. But if that's true, then you have to assume compositionality. Somehow you have to assume that you can decompose that object into parts such that that particular cell could be activated both for concept 1 and concept 2. So, two questions. First of all, are these only individuals? And then for your model, must you assume compositionality of your concepts?
So it's not just for individuals. It is for whatever is relevant for the subject to potentially create a memory. I'll give you an example. It doesn't fire, I never found a neuron firing to a chair or to an orange or something like this. I did find a neuron firing to guacamole, and that was because it was a Mexican girl that couldn't take the hospital food anymore and was dreaming to have Mexican food again. And for her it was very relevant guacamole, and I showed a picture of guacamole and I said, boom, I got a neuron firing, to top. So it is for, it can be whatever. Spaghetti, yes. So now, but you never find neurons firing to faces, like in cortex. And why is that? Because if I see you, I'm not seeing you, a male person that is a scientist, Italian, so I'm just seeing you. So we tend to individualize, and we tend to form memories individualizing people. Of course, you can then say, well, I know that you are an Italian, you're a scientist and so on, but I think this is all information that is cortical. So that's why we individualize. And I think you categorize at the level in which you want to store the memory. You can store a memory, there is an animal passing by, or there is a Labrador, or no, this is actually my dog. So how you want to categorize, well, it's depending on the meaning that it has to you. I'm not sure then I understood the second part, but basically if you have conjoined features and things like that, I think all this is resolved in cortex, and then you extract the meaning in cortex, the meaning is extracted in cortex, and this is what is fed into the hippocampus. So whatever it is, it may or may not make some neuron fire. Now, if you create an association, if you do these conjoined features, it's not that you will have new neurons encoding for the association, because you run out of neurons very quickly if you do that. It's not like I think the standard HEP model. So you have representations for concepts, and what you do is create overlaps. That's my idea. No, I have no proof.
So what I was referring to when you showed us the very beginning of the slide, you had the network for Skywalker, and then you had Yoda, and then you showed how one of those cells responded to the other object. Yeah. And the question was, why only that cell? Why not all of them? So if only one of them responds, presumably, perhaps by assumption...
It's a cartoon, no? Say you have 10,000 Skywalker neurons, 10,000 Yoda, maybe 500 will start firing to Yoda. But they cannot be all because...
Why? Why those?
Because then you cannot distinguish them anymore. So if it is a full overlap, there's nothing that tells me which of the two it is. And a full overlap is what happens, for example, what I said before, when you link a face with a name. And that's why we have, where when you say the name, you show the face, for the neuron it's the same, because you have a full overlap.
Okay, first we have a question in the back.
I have a question about the finding that if the cell fires, does the subject see the stimulus? If it doesn't fire, does the subject fail to see the stimulus? If we look at it from the perspective of signal detection theory, then both the cell and the participant are faced with a decision problem under uncertainty. So there's noise in the system. The noise could drive the cell above threshold and also the observer above threshold. And if the noise is positioned before the decision problem of both the cell and the observer, then the responses should be correlated.
So I think I will put your argument, the starting of your argument, the other way around. It's not that, how you say it? It's not that the cell is involved in the perception of the stimulus. The perception of the stimulus is done elsewhere. And this neuron is just getting the output from cortex. Perception is not happening here. And basically, in cortex, you may have more graded response, distribute coding, robust to noise, and so on. And at this level, there's no computations done. It's basically getting the result from cortex. They say, it was Jennifer Aniston. Maybe it was not. Well, I believe it was Jennifer Aniston. That's it, period. And if it was ambiguous or not, I believe it's Jennifer Aniston, and therefore I fire. So, but all the decisions of what it is and so on, they're in visual cortex.
I think that even if there was only noise in the system, the two levels would be correlated. If it's only noise that carries the decision process over a threshold to a yes response, then if the noise is affecting both the cell and the entire observer, because it's early in the system, then the two responses have to be correlated. And they will both respond alike, the cell and the entire person.
Yeah, but this is because the noise is biasing the decision than in cortex. I put another example, more brutal. I close my eyes and I think of Luke Skywalker, and my Luke Skywalker neurons are firing. So we also show that. They can't even know stimulus. So it's basically what the subject believes he's seeing, or what the subject is thinking, for whatever reason.
Okay. Sharon,
Thank you. I'm trying to put together this partially overlapping assembly idea with everything I thought I knew about what the hippocampus is doing. And there's a few places where I'm struggling. One arises from what I thought was a very surprising fact about your data that you relayed, which is that if one of your cells fires to multiple concepts, that those concepts are related. And one of the things I thought I knew about why we need both the hippocampus and cortex is that in cortex, the nature of information coding there is such that relationships are those population codes are structured such that long-term relationships are present, but not so in the hippocampus. And why not so in the hippocampus? Because now if I want to learn an arbitrary association of Yoda and the White House, I really need to separate the Yoda pattern from the Luke Skywalker pattern so that I don't accidentally think of Luke Skywalker in the White House. And so having non-overlapping representations of similar information I thought would be pretty important. So I was thinking this is a big problem until you just answered that last question. If you get these effects with your eyes closed, then it seems like your association effects that you mentioned earlier could just be that when someone says Yoda, you think Luke Skywalker. It's like the eyes closed Luke Skywalker, in which case you don't need partially overlapping assemblies at all.
So when I say explicit meanings, you can see it with single cells, but the meaning or the awareness is given by the population. So basically, when I show this cartoon, and if I show you Luke Skywalker, well, you also have some Yoda neurons firing, but you're not confusing those because you have nearly the whole assembly of Luke Skywalker fire and not that many of Yoda.
I got that, but now you say White House, and my White House neurons don't know to go get the, Yoda neurons that aren't connected to Luke Skywalker, right? So some of them are going to combine with Yoda. What if it happens to be the Luke Skywalker ones? Yeah?
I understand it, sorry.
But it's like a Venn diagram that now has 3 things instead of 2. And so some of the White House will overlap with Luke Skywalker also.
Yeah.
Right. So that's what I'm saying. That's a problem. That's a problem for memory.
Why? I mean, you have different neurons encoding different overlaps. I mean, you can keep on creating associations, and then you keep on creating overlaps.
It's a problem for creating separations such that White House doesn't bring to mind Luke Skywalker. If now White House is partially overlapping with Luke Skywalker. Like imagine 3 circles in a Venn diagram, and there's going to be some overlap now of White House and Luke Skywalker. And that's a problem. So it seems that if you're when you learn an association between A and B, you really need to separate. I mean, this notion of pattern separation in the hippocampus is a very dominant idea right now in the field.
In rats, not in humans.
In humans, there's...
It's fMRI. I don't think there's pattern separation in humans as in rats. I don't see it because pattern separation, for example, you have the place self remapping, right? You change the context, and you have a completely different pattern of firing of place cells. I don't see this here. I mean, I can show the picture of Jennifer Aniston or a movie or The Voice or whatever. I mean, in whatever environment, in whatever context, the neuron fires. And I think these points, I don't know, this is a theory, and I'm speculating, but it's a discussion session, so I mean, I hope you don't mind. So it's plainly speculation, no? I'm hoping to get data to prove this, but I still don't have it. But I think this is the main distinction between humans and rats, that for the rat it's very, very hard, for the rodent it's very, very hard to abstract things, to know that this place is the same. If I move, if I have the maze here, if I have it here, it's the same place. I mean, they completely remap everything. For us, it's obvious, no? If I see you in another conference, you're still the same person. You're not a different cell assembly encoding another thing. So that's why I believe pattern separation, there might be some pattern separation in humans, but not as strong as in hippocampus, because we do want to have abstraction as well. So we do want to know that a concept is the same in different contexts, but it's speculative.
I thought that's what cortex was for.
So my question is kind of a follow-up of one that was asked earlier, and kind of Sharon's too, I think, which is, to what extent do you think these concept cells might be more like, instead of identity cells, more like verbal label cells? So the activation you were talking about starts at about 300 milliseconds after the onset of the picture, which is just enough time to activate the verbal label, and probably all these things that are important to us we have verbal labels for. So I'm wondering whether, and it's hard to get people not to label things that they know. I think probably especially in a context like this.
But sometimes you do get responses, I don't remember one right now, but you do get responses to people that they don't have the name at hand. They don't remember the name, but they know the person. Maybe you know this is a famous actor, I know this guy, and then you get a neuron firing to the guy, but we still not come up with a name.
Right. Although you can have the name, I mean, we know from production that you can have the name on the tip of your tongue, so you can have some activation of the name. So I guess I'm wondering whether you could do something like teach people new things that you manage to teach them without verbal labels. It's hard. You have to do some kind of verbal interference or something like that, but whether you would also see the same kind of
Well, ideally you would like to have pictures of people they know but they don't remember the name, or they don't know the name. They don't know the name at all.
Yeah, yeah,
but
yeah.
I don't have data for that.
Ori, did you have a question? Yeah.
I don't think this came up. So in this kind of example with Luke Skywalker and Yoda, does it ever happen that there's a third thing it fires for? Say, for example, Star Wars or Princess Leia.
Does it ever happen? What, sorry?
So when you've got this cell that's firing for both Luke Skywalker and Yoda, does it ever happen that it fires for a third associated thing?
Yeah.
For example, like Star Wars.
Yeah. I mean, I have neurons firing to me, I mean to myself, and to two other guys doing experiments. So, I mean, the overlap doesn't need to be only constrained to two concepts.
I guess that's an alternative interpretation, and maybe this is ruled out by lots of things I don't know about. Could it be that it's actually a Star Wars cell? And then the reason it's not that it's not an overlap between them cells, it's simply a Star Wars cell, and since both of them would somehow be subsumed under that concept.
Yeah.
Or if it was a cell for the relationship, between the three of you.
Yeah. In many cases unlimited because, I mean, the way to prove that it's not would be to try more characters of Star Wars. With the Luke Skywalker cell, I didn't have that. But I did have it with the Jennifer Aniston neuron. Actually, the Jennifer Aniston neuron didn't fire to Jennifer Aniston, fired to Rachel, which is the character Jennifer Aniston played in Friends. And it didn't fire to the name Jennifer Aniston. And I tested next day. I mean, when I saw this neuron firing, I said, well, what is this neuron doing? So I tested, and I showed pictures of all the characters of Friends. And it fired again to Jennifer Aniston, and it fired to Lisa Kudrow, but not to the other characters of Friends, and not to the logo of the TV series. So we have many, many cases like that.
Okay.
Okay, last question here.
So I guess we're all interested in what we think, or you think, the causal role is of these cells. And I'm just wondering, why are they, going back to all these concept cells, are they not experience cells? Are they not telling us about what the current episode is? Because what I don't know from this work is if the patient goes out of the situation where you're presenting this on a laptop in front of them in that hospital, it just tells you about the UK health service hotel. If they now walk outside and they sit on a bus where the context is now different, will I get this same experience? Because it could be that this is actually context-specific, episodic activation. It's the equivalent of a variable that I'm instantiating for the purpose of the current context and may have some causal role in subsequently being able to refer to this particular context in which you are the experimenter, blah, blah, blah.
Yeah. To answer the question, I mean, we had to wait. I mean, we're doing exactly these experiments right now. I'm limited, no, because I cannot bring Jennifer Aniston. I think she wouldn't agree to come. I cannot bring these people, or I cannot, but the experiments we're doing, we're trying to change the context at least as much as we can within our experimental setup, and to see if what happens is more like in rats, that you get pattern separation, or if it is more like an invariant representation, irrespective of content. My gut feeling is that it may change, but not as much as you will see in the rodent hippocampus. My gut feeling that it will not change much is I'm limited, no? I'm still showing pictures and things in my laptop, or the sound and so on. But it's in completely different experiments. So, but you're right. I mean, it's still seeing this person doing an experiment in hospital. So, I mean, I'm limited in this sense. Yeah.
Okay. Thank you very much.